Today we shall be discussing in very brief about glands. What are glands? Glands are specialized secretory cell or cells derived from the epithelial tissue and elaborate secretion. What is the function of glands? Glands, like the endocrine glands, they secrete different hormones. The mucus secreting glands will give rise to mucus. The sweat glands of the skin secrete sweat. The sebaceous or the oil glands of the skin, sebum. Saliva from the salivary glands of the mouth. Cerumen or earwax from the ceruminous glands of the external auditory canal. Bile from liver, digestive enzymes from the pancreas, and milk from the mammary glands. How do glands develop? Now the glands, they develop as an invagination of the epithelial cell lining into the underlying connective tissue. So the epithelial cells invade into the underlying connective tissue, they multiply, and then they get reorganized. The distal portion of the glands, they form the secretory end piece, that is the functional portion, which will secrete the secretions. And the proximal part, it forms the duct through which the secretions are conveyed to the surface. This is the development of the exocrine glands. In case of endocrine glands, the connecting portion, which was connecting the secretory end piece with the surface, this connecting link disappears. Hence, the endocrine glands are the ductless glands. This is the general architecture of a gland. You can see this brown portion. This is the duct portion. Over here, the ducts have undergone branching. And this blue color portion is the secretory end piece, which will give rise to the different secretions. In some glands, we have special cells known as the myoepithelial cells. These myoepithelial cells are present between the basement membrane and the secretory end piece. And as the name suggests, these are cells which have the characteristics of both muscle cells as well as epithelial cells, and they help to express the secretions into the ducts. This is a picture of the myoepithelial cell surrounding the secretory end piece. Now, classification of glands. How do we classify glands? It can be classified in different ways, like according to histogenesis or development, according to site of secretion, according to the number of cells, the number of ducts, the shape of the secretory end pieces, and according to the nature of secretion. So we will discuss them one by one. Now, classification according to development or histogenesis. We have ectodermal glands, mesodermal, and endodermal glands. Ectodermal glands are glands of the skin, the mammary glands, lacrimal and salivary glands, and the pituitary gland. The mesodermal glands are the kidney, spleen, the gonads, that is the testes and ovary, and the suprarenal cortex. Endodermal glands examples are the thyroid, parathyroid, thymus, liver, pancreas. We have the glands of the alimentary tract, the respiratory tract, etc. Now, classification according to site of secretion. We have three types: the exocrine, the endocrine glands, and the paracrine glands. Exocrine glands, as we have already discussed here, the secretions are conveyed to the surface by means of a duct. In case of endocrine glands, the secretions directly is conveyed into the bloodstream. In case of paracrine glands, the secretions are conveyed into the local extracellular space, and here it has action in the surrounding adjacent cells. So the basic difference between exocrine glands and endocrine glands. In case of endocrine glands, the secretions are directly liberated into the bloodstream, and it has no duct present. So the endocrine glands are also known as the ductless glands. In case of exocrine glands, here we have ducts, and the secretion is conveyed to the surface of the epithelium by means of these ducts. This classification according to the number of cells. We have unicellular gland that is composed of a single cell, for example, goblet cells of the respiratory and intestinal tracts. And we have multicellular glands that is composed of many cells. As we all know that the goblet cells are present in the lining of the respiratory and intestinal tract, and its product is mucin. The mucinogen granules containing the mucin, they are present in the apical part of the cell, and the nucleus is pushed towards the basal side, And this goblet cells, they have a shape of a drinking glass, so its name. As we all know, in HE staining, the goblet cells, they have an empty appearance, and it protects the lining of the intestinal epithelium, and also to trap the foreign bodies and bacteria in the respiratory tract. Next is classification according to the shape of the glands. If the secretory end piece, it appears tubular, straight, or coiled, it is said to be tubular variety. If the secretory end piece is alveolar, flask shaped or rounded, it is known as the alveolar or acinus variety. And if it is a combination of both tubular and the alveolar varieties, it is known as the tubulo-alveolar variety. Next is classification according to the number of ducts. If an exocrine gland has a single duct, it is known as simple gland. And if the exocrine gland has a duct which undergoes repeated branching and has thus major and minor ducts, it is known as a compound gland. In this type of classification, according to the number of ducts, whether it is unbranched or branched, we may have the following different varieties. These are simple glands, without duct, with duct, simple branched tubular, simple coiled tubular, simple acinar. Next, according to the branching patterns, it may be a complex gland, complex or branched tubular, branched acinar, and branched tubulo-acinar varieties. Next classification is according to manner of secretion. We have three types of glands: the merocrine glands, the apocrine glands, and the holocrine glands. In case of merocrine glands, the secretions are present in vesicles bounded by the membrane. The membrane of these vesicles will fuse with the apical plasma membrane, and the secretions are liberated to the exterior. That means the secretions are conveyed to the exterior by means of exocytosis. Example, the salivary glands. Next is the apocrine variety. Here, the apical part of the cytoplasm of the cells is lost along with the secretions. Example is the lactating mammary gland. Next is the holocrine variety. Here, the whole cell bursts to liberate its secretions. So there is loss of whole cell. Here, the cell which is lost is replaced by a new cell. Example is the sebaceous glands of the skin, which secrete sebum. Another variety is the cytocrine gland. Here, cells are released as secretion. Example, the spermatozoa from the testes. Next classification is according to nature of secretion. We have three types: the serous glands, the mucous glands, and the mixed glands or the seromucous glands. The serous glands, they secrete thin watery material which is rich in enzymes. Example is the parotid salivary gland. The mucous glands, on the other hand, secrete thick viscous material for protection and lubrication. Example is the sublingual salivary gland. The mixed gland, they have both serous and mucous acini. They secrete watery and viscous material from both the serous and the mucous acini. Example is the submandibular salivary gland. This is a serous diagram showing a serous acinus, mucous acinus. The serous acinus, as you can make out from the diagram, they are smaller in size compared to the mucous acini. In case of serous, the lumen is smaller. In case of mucus, the lumen is larger. We can see that the serous acini, it takes dark stain, and the nucleus are central and rounded. On the other hand, in case of mucous acini, it takes a pale stain, and the nucleus is towards the basement membrane. This is a table showing the difference between the two. We can see that the secretory end pieces are small in case of serous acini, large in case of mucous acini. The nature of secretory granules is, in case of serous acinus, is zymogen granules, which has enzymatic action. In case of mucous acini, mucinogen droplets, which help in protection and lubrication. The serous acini are darkly stained with small lumen. The mucous acini are lightly stained with large lumen. The secretion of the serous acinus is thin and watery. The secretion of the mucous acini is thick and viscous. Example of the serous acini is the parotid gland, and that of the mucous acini is the sublingual gland. Now, general architecture of the glands. Most of the glands are composed of either serous or mucous secretory cells, or both. These cells, they form secretory end pieces, which are either flask-shaped, that is acini, or cylindrical, that is tubules. The end pieces are often associated with contractile myoepithelial cells, whose function is to express the secretion. The secretory end pieces, they form the parenchyma, and the connective tissue framework of the gland, which supports the parenchyma, it forms the stroma. So glands, the architecture of the glands consists of two things. One is the parenchyma and the stroma. The parenchyma, it consists of the secretory end pieces and the ducts, which may be intralobular, interlobular, and the main excretory ducts. The stroma is formed of loose connective tissue, which forms the structural framework of the gland. It is composed of capsule and septa, like the interlobular and the interlobar septa. So this is the general architecture of the gland. We can see these are the lobules, and they are separated by the interlobular septa. And in between two lobules, we have an interlobular duct. And inside a lobule, the duct present is the intralobular duct. This is a picture of the serous salivary gland. We can see these are the different lobules, and we can make out the darkly stained serous acini with small lumen and rounded nuclei centrally placed. And in between the lobules, we can make out the ducts. In this slide, we can see the mucous gland, that is a sublingual salivary gland. We can make out the pale staining mucous acini with nuclei which are flat and basally located. And we can also make out these cluster of cells, these semilunar cluster of darkly stained cluster of cells over the mucous acini. These are the serous demilune. These are mixed glands. We have both serous component and mucous component. And it is basically an artifact where the cluster of serous cells are seen attached to the mucous acini. This slide is showing the mixed gland, that is the submandibular salivary gland. We can see both serous acini and mucous acini. We can differentiate the two from their staining appearance and also from the characteristics which we have learned earlier. And we can also make out the serous demilune. Glands can thus be classified into two groups, the exocrine glands and the endocrine glands. The exocrine glands can further be classified as unicellular and multicellular. Multicellular glands is further classified as, according to the shape of the glands, we have number 1 tubular, 2 is the alveolar variety, 3 is the tubulo-alveolar variety. Then according to the ducts, it may be simple, complex. According to manner of secretion, it may be holocrine glands, apocrine glands, merocrine glands. According to the type of secretion, it will be serous acinus, mucous acinus, and the mixed variety. And according to development, it will be ectodermal, mesodermal, and the endodermal glands. Then endocrine glands further are classified as cord and clump variety, and the other is the follicular variety. Let us discuss in very brief about the endocrine glands. The endocrine glands, as we already know, are ductless glands which secrete the hormones and other useful substances directly into the bloodstream. And the glands may be of two types. One is the cord and clump type, the other is the follicular type. The different endocrine glands of our body include the hypothalamus, the pituitary, the thyroid, parathyroid, the adrenal glands, the kidney, the pancreas, the gonads, the testes and the ovary, and the thymus. In the cord and clump variety, here the cells are arranged in the form of irregular cords and clumps, that is in clusters, and the secretion is stored within the cells, that is intracellularly, and when the body requires these hormones, the secretions are delivered directly into the bloodstream. Most of the endocrine glands belong to this variety. In the follicular variety, here the cells, they secrete extracellularly and store their secretion in the form of follicles. That is, they form a pool of secretion and which is surrounded by the follicular cells. Example is the thyroid follicle. This is a slide showing the thyroid follicles. This is the secretion of the thyroid follicles, that is the colloid, and it is surrounded by the follicular cuboidal cells. The follicular arrangement of the thyroid gland helps to keep the thyroglobulin of the thyroid follicles as a secluded antigen from the body proteins. Thus, the body proteins cannot initiate an immune response against this antigen. Hashimoto's disease is a type of autoimmune disease. Here, antibodies are formed against the thyroglobulin. As a result, the lymphocytes rich with antibody, they aggregate in the interfollicular spaces. This causes compression of the follicles and diminished function of the gland, resulting in hypothyroidism. Next is adenocarcinoma. These are tumors arising from the glandular epithelium. It is commonly seen in the colon cancer, in stomach, uterine endometrium, etc. Thank you.